Keeping in mind again that when we spin down blood, we end up with erythrocytes at the bottom, plasma at the top, and that buffy coat in the middle. And remember what the buffy coat is? Buffy coat is made of the leukocytes, which we just discussed in the previous video, and then the platelets, which we will be discussing now. Okay, so it's helpful to look at the process of platelet formation. This is called thrombopoiesis. It is enacted by a particular hormone, which is called thrombopoietin, which is developed in the liver and the kidney. Okay, so platelets tend to be quite small. They are considered to be cytoplasmic fragments of megakaryocytes. So here we go. The platelet itself is about 2 to 4 micrometers in diameter, but it's sloughed off of a much larger structure called a megakaryocyte, which is quite large, 60 to 100 micrometers, one of the largest cells in our body, actually. Take a look at this. When we think about the hematopoietic stem cell, which eventually derives our platelets, that is the same structure, the hemocytoblast, which, remember, it gave rise to red blood cells and white blood cells. So in this case, the hemocytoblast will differentiate in the presence of thrombopoietin. All of this takes place in the red bone marrow, as is true of all of these developmental processes. As you can see, the hemocytoblast ends up becoming several mediaries of it before it becomes a megakaryocyte. The megakaryocyte, as the name indicates, karyo indicates nuclei. So there's many nuclei inside of here. It's basically a cell that became enlarged and continued to undergo mitosis without actually undergoing cell division or cytokinesis. So it ends up being this large, massive cell with many nuclei inside of it. The way that it works is that the megakaryocyte basically sticks out long tendrils of its cytoplasm into the blood vessels, and it kind of splits off fragments of itself, and that's how platelets are born. You can actually see some of the platelets sort of surrounding this megakaryocyte. So these loose little structures that are able to leave the megakaryocyte easily. So one of the preeminent features of a platelet is that it has these granules that aid in your blood clotting. What's inside of here? These granules include serotonin, calcium, enzymes, ADP, this special stuff called PDGF, which is platelet-derived growth factor. All of those chemicals will aid in clotting your blood, which we'll be talking about next. The platelets can circulate freely. The point of a platelet is to form temporary plugs in damaged capillaries. And of course, they are anucleate. There is no nucleus inside of here, so they are not considered actual cells. They're considered formed elements. They tend to degenerate in about 10 days, maybe 5 to 9 days or less.